All right, part three here. I'm going to talk about some few more specific details about some example viruses, just to see some of the variety and how viruses replicate themselves. They all have their own little quirks. First group I'm going to talk about are the herpes viruses. There are lots of different kinds of herpes viruses, including the ones that cause genital and oral herpes. But then your chickenpox virus is a type of herpes virus. The virus that causes mononucleosis is a type of herpes virus. There are a number of herpes virus cousins. This diagram shows you what herpes viruses have to do. These are they're a double-stranded DNA virus. That means they have DNA inside the capsid shell. They have an envelope. They attach to the host cell receptor, and then they do fusion, membrane fusion. We talked about that on the last video clip. The capsid comes inside, and then it actually attaches to fibers of the cytoskeleton, slides along those. It goes through an opening into the nucleus, a nuclear pore, winds up inside the nucleus. Now finally, the virus's DNA comes out. So your viral DNA genome winds up in here. And early and late messenger RNAs there on the diagram. That's just referring to, okay, so herpes virus, the DNA has genes along the genome, and those undergo transcription. You make messenger RNAs, and some of them are transcribed earlier during the replication cycle and some later. Those messenger RNAs go out of the nucleus, just like our messenger RNAs do, out here into the cytoplasm. You make new viral proteins, protein synthesis. Those are supposed to be ribosomes that you see there. Proteins of the virus come back into the nucleus. The virus's DNA gets replicated inside the nucleus in here. It's hijacking some of the natural normal processes that would happen inside the nucleus. You assemble new capsids. So the capsid shell protects the new DNA genomes that were made in here. Those come out of the nucleus. And then you have budding that takes place, but the budding actually occurs in the endoplasmic reticulum. If you remember that from earlier courses, the endoplasmic reticulum is a membrane-covered compartment. Inside our cells, sometimes called the ER. So it's a membrane-covered compartment, and these herpes viruses actually push themselves into that compartment. So through the membrane, and that's where they're actually budding. They pick up their envelope by budding into the ER. And then if you've learned about the ER before, you've learned that things in the ER go to another compartment called the Golgi apparatus. I can't write with my little stylus here, it's not cooperating. And then they go to the cell surface and they get secreted to the outside, like they're showing you there. And that is like a natural assembly line process that takes place inside our cells. Proteins that are going to be secreted to the outside, or that are going to wind up in the cell membrane, are first manufactured in the endoplasmic reticulum. Then they go to another compartment called the Golgi. And then from there they get sent to the cell surface to either be secreted, or they wind up staying in the at the cell surface. So for herpes virus, it's actually acting like something that this host cell would naturally want to secrete, like a hormone. So it's hijacking the normal processes like a hormone would use to get out of the host cell to have itself exit. So those are some little quirks with herpes viruses. So they're kind of odd. Instead of budding at the host cell membrane, they wind up budding into the endoplasmic reticulum. Okay, negative strand RNA viruses or minus strand RNA viruses. Either of those terms can be used. Good example, influenza, an example of one of these types of viruses. So it has, its RNA genome is negative polarity. My pen doesn't want to write. There we go. I guess I'm off the surface. which means when it replicates, you got to go from minus to plus and then make new minus strands, like we talked about before. Also, the plus strands are the ones that the host cell will see as mRNA, so you've got to make those as well. And flu viruses have their own RNA polymerases that do that and go from minus to plus to new minus strand RNAs. All right, and again, that kind of enzyme that's copying RNA, going from RNA to more RNA, that's not something that our host cells do. Our host cells go from DNA to RNA, not RNA to RNA. So those enzymes are unique to RNA viruses, so if we can come up with drugs that will inhibit those enzymes, those would be good things maybe to use in the future to treat patients with. Influenza is interesting because if you look there inside the virus itself, the genome is actually divided up into 8 pieces or 8 segments of RNA. Those different segments have different genes on them. So it's almost kind of like our DNA is divided up on, among different chromosomes. The RNA of influenza virus is divided up into different pieces as well. So different genes are on different segments or pieces of RNA. Now, interestingly, I'm going to erase all of that. The fact that influenza's RNA is divided up among those 8 pieces is one of the reasons why influenza virus can undergo some pretty dramatic changes. Because, for example, let's say every once in a while, there are flu viruses that infect humans, flu viruses that infect pigs, flu viruses that infect birds. Well, every once in a while you'll have a respiratory tract cell in a pig or a human or a bird that gets simultaneously infected with 2 different kinds of flu. So let's say you got a host cell and in comes a swine type flu, 6, 7, 8. There's its 8 pieces of RNA, and then at the same time it gets infected with some other type of flu, 3, 4, 5, 6, 7, 8 pieces of RNA in blue, and all of these are replicating inside that host cell, and you start packaging up new flu viruses. Well, guess what? Sometimes you wind up, not sometimes, but pretty frequently, you're going to wind up with new viruses that might have, let's see, 3, let's say 5 RNA segments from, say, a human flu, and then 1, 2, 3 from a pig flu. And all those wind up getting assembled together into a flu virus particle. That flu virus particle gets released, and now it's free to go out and start infecting new host cells. Well, every once in a while you have some sort of a rearrangement like this that creates a new strain of flu that is maybe more deadly or more pathogenic than earlier strains. Also, it can be a new strain that the human population does not have much immunity to yet, so it winds up causing a big-time epidemic. You know, maybe around the whole world it might become a pandemic. So because our influenza has its RNA divided up onto those multiple segments or pieces, that's one of the reasons why it has been such a troublemaker and it changes so rapidly. That's not the only reason. The other reason is because its RNA polymerase enzymes are very prone to making mutations. They make errors pretty frequently, and sometimes those errors are beneficial for the flu, and it winds up being able to escape the immunity that the human population has developed through exposure or vaccines. So influenza is pretty crafty. Oh, and by the way, the H1N1 flu from a few years ago that everybody was so scared about, if I remember correctly, the index case for that was a farmer in Mexico, and in one of his cells inside his respiratory tract, was simultaneously infected with, there was one flu that came in that's a bird swine flu combination. So it had already done some hanky-panky swapping of RNA earlier. And that cell also was infected with a human flu strain. And those two got together and shuffled their little pieces of RNA, and out popped the H1N1 strain that started spreading from there around the world. Or it may have been that the pig was the index case and a farmer picked it up from the pig, and from there it started spreading around the world. Here's a nice little diagram which gives us some more information on HIV, human immunodeficiency virus, an example of a retrovirus. And again, retro, we're going backwards. So these are viruses that have RNA genomes, but they take those and convert them to double-stranded DNA after they enter their hosts, and that process is called reverse transcription. Stylus is not cooperating. Okay, so if we take a look at what's going on over here with HIV. So here's an HIV virus particle. We've already talked about how it has these spikes in the surface called GP120. Those attach to a host cell receptor that's called CD4. You have that on your macrophages, especially on some of your T lymphocytes, a type of white blood cell that's very important for your immune system function, and also on a few other cell types. There's also on this diagram, they're showing you a co-receptor. There is another receptor molecule that HIV also binds to, and some strains of HIV use one called CCR5, and then others use one called CXCR4. Those are also molecules you find in the surfaces of those types of cells I was just mentioning. So HIV attaches to both of those. Once that happens, notice the capsid here, which is kind of shaped like a little bit, almost like a funnel a little bit, comes into the host cell. There's reverse transcriptase. So it winds up taking the RNA genomes, or actually two of the RNA genomes inside the HIV virus capsid, converts that over to double-stranded DNA there in yellow. Then there's an enzyme here, if you see this one, integrase. What it does is it cuts your DNA, the host cell, hopefully not your DNA, but the host cell's DNA, and inserts, pastes in the HIV DNA. That's called integration. Once that happens, the host cell now just thinks that HIV DNA is some of its own DNA, so it's going to start doing transcription of HIV's genes. Those RNAs go out into the cytoplasm, and the ribosomes are going to see those and make the virus's proteins. So you assemble new virus particles. Some of these RNAs are also going to be the new genomes, because we have RNA genomes inside HIV. You assemble all of those things together, and then HIV pushes itself through the host cell membrane. It buds at the cell surface, and eventually we have a mature HIV virion. So then that's able to move along and spread and find new host cells. One of the reasons why HIV has been so difficult to treat, or one of the reasons why even when patients are on drugs that are keeping the virus out of control, if they go off of those drugs, the virus comes right back. Once HIV has done this, it has stuck its DNA inside its host cell. You know, it may go, you may be treating the patient with drugs, and these types of processes over here, the virus assembling itself, or new viruses starting infections and doing reverse transcription, those types of things may be screwed up, but you still have long-lived white blood cells in the body that are called memory cells, where HIV has inserted its DNA. So it can just be kind of sitting there in a latent state or quietly. And if a patient goes off of those drugs, all of these processes you see on here just resume. So those genes right there start undergoing transcription and translation, and you just start the whole story over again. So that's one of the reasons why HIV has been such a pain to try to combat. So those were just a few example viruses. There are many, many, many others. So just kind of a summary about how viruses replicate themselves. All viruses have to enter a host. They're going to hijack the host cell's ribosomes to make their proteins. And they've got to exit the host in some way. And that's either through lysis, that's breaking apart the host cell, or by budding, pushing itself through a cell membrane. How do viruses differ from one another? By the host cell receptors they use, by their method of entry. Is it direct penetration, membrane fusion? Do they use endocytosis to get in? They differ by having RNA or DNA genomes. They can differ in how they replicate those genomes. Some RNA viruses are plus strands, some are minus strands, some are those retroviruses. And then, it even gets down into some of these nitty-gritty details that we've been looking at where, some are in the nucleus replicating themselves, some are out in the cytoplasm. Some envelope viruses will bud at the inside the cell in the ER, and some are at the cell surface pushing themselves out. So lots of variation that occurs with viruses. A couple of other things about viruses, cytopathic effects of viruses on their host cells. So we've said before that when you have an infectious disease, you don't have a disease unless you're being harmed in some way. Well, many viruses do cause nasty things to occur in their host cells, and those are called cytopathic effects. For example, some viruses are going to cause their host cells to lyse, break apart. Some enveloped viruses actually do this weird thing where they cause their host cells to fuse together and form giant cells. Those are called syncytia when that occurs. So like in this diagram over here, right there, kind of outlining with the cursor, there's a normal single cell, and over here you have this ginormous cell. In purple, there are all those purple spots are nuclei, and then that's all one cell. Well, what happens is you get all these enveloped viruses budding from the surface, and they attach to a neighboring cell, and then because those viral envelopes and their host cell membranes can all melt together, it causes these adjacent cells to fuse together, and you wind up with these big giant cells that develop, and those are called syncytia. And that can often be observed like in the laboratory if you're working with viruses. Some types of viruses, like HIV, for example, is an envelope virus that makes these syncytia. And so if you can grow the T cells the virus likes to infect in the laboratory and put HIV in there, and it starts going inside the T cells to replicate. And if you look under there after a few days, you'll see these big, gigantic syncytia where T cells have fused together as the HIV virus has spread around. Other viruses will set up, this is a host cell for a virus, and these green areas you see on the inside are called inclusion bodies, and that's where some viruses will set up these elaborate viral replication factories inside their host cells and take up quite a bit of the space in there. Those are called inclusion bodies. Some other examples, like with poliovirus, for example. Poliovirus is actually still studied quite a bit because it's a pretty good model virus. It has a lot of relatives. Poliovirus is not a medical problem in the United States anymore. It is still in some parts of the world at low levels. But it has lots of close relatives, including rhinovirus that causes the common cold is a good example. Foot and mouth disease is another good example of a close relative of polio. They don't cause the severe type of deadly infections that poliovirus causes, but the way they operate is very similar. If you were working in a laboratory, for example, over in our biotechnology laboratory in Huntsville, we can grow human cells as host cells for viruses, and you tend to grow them in little plastic containers like this, and they grow as a sheet. The cells are living as a sheet on the bottom of the flask, and that red fluid you see there is providing, it's like a broth, it's providing nutrients to those cells. So if you were looking at host cells for poliovirus, under the microscope growing in a little container like that, they would look like this. So there's one cell, there's another. You can see the nucleus in the center. Those are healthy looking host cells. And over here, this is what they look like if they're infected with poliovirus. They tend to round up as the virus takes over most of the machinery inside the host in order for the polioviruses to replicate themselves. They ball up and look really, really sick, and then they die pretty quickly, usually within a matter of 24 hours or less. They're dead, and then they're floating around in the fluid inside the flask. So that's another example of cytopathic effects. If you read the Henrietta Lacks book about the HeLa cells, poliovirus loves HeLa cells as a host, and so it will do nasty things like that to those types of cells. How do we detect viruses? We're not as good at diagnosing viral infections as we are at diagnosing bacterial infections for several reasons. One thing, you can't visualize them with typical light microscopes. You've got to have very powerful electron microscopes, so you don't do things like gram staining on viruses. Not going to work. They're way too small. Also, culturing them is not, you can't put viruses on nutrient agar or any of those slants or lactose fermentation broth or any of that type of stuff like we used for your unknown bacterium project, because viruses need host cells in order to grow. So human pathogenic viruses typically need human cells as hosts to grow in the laboratory. And growing human cells in the laboratory is a lot more expensive. It's more technically challenging. People have to be better trained. It's not quite as easy to do. You need lots of expensive equipment to make that happen. All right, and so because they're more difficult to grow, the typical clinical laboratory in a hospital is not equipped to grow them, to study them, or to diagnose viral infections. But we're getting better. PCR, like we talked about, that very sensitive genetic-based technique, more and more that's going to be used to help diagnose viral infections in patients. And then there also are assays called immunodiagnostic assays. These are assays that take advantage of immunos, referring to the immune system. And when you study the immune system, you learn about antibodies. These are protein weapons that the immune systems of animals like us make, and they specifically tag very, very specific molecules on things that are foreign to the body, like bacterial molecules, viral molecules. And so those can actually be prepared and used as tools to very, very specifically detect very, very specific things. And they're also very, 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 very sensitive. And assays that use those are called immunodiagnostic assays. And we're going to see an example of that as we go over the immune system. We'll actually do home pregnancy tests in the laboratory as an example of one of those types of assays. We'll also do blood typing. That's another example of an immunodiagnostic assay where you use antibodies as tools to determine blood type. Or if you've ever had a rapid strep test, that's an immunodiagnostic type assay as well. So there are some pretty good examples of those that can be used for some types of viral infections also. Another last little interesting thing about viruses, there are viruses that cause cancer, and they do that by either bringing in a gene into the host cell that's called an oncogene. Like an oncologist is a cancer doctor. An oncogene is one that when it's expressed, it triggers uncontrolled cell growth to happen. So it's a cancer gene, because when a cell starts dividing in an out of control manner, that cell can become cancerous. Or sometimes those proteins are involved in preventing a cell from dividing in an out of control manner. And if you mutate the gene that codes for that protein so that it doesn't work anymore, those proteins can kind of function like gatekeepers, and if they're not working, the cell gets past those gates and starts dividing when it's not supposed to be dividing. So sometimes viruses will bring in their own oncogenes, and they'll make virus proteins that causes the cell to become cancerous. In other cases, viruses will mutate host cell genes or convert them into oncogenes and cause the host cell to become cancerous. Probably the best known example of a virus that can cause cancer, human papillomavirus. There are certain strains that are cause cervical cancers. That's HPV. And as you know, kids are being vaccinated now against the few strains that are known to be the most involved in cervical cancers. There's another type of virus, HTLV, human T lymphotropic virus type 1, which can cause a T cell type cancer. It's actually a relative of HIV. Epstein-Barr virus, that's your mononucleosis virus. But it can trigger a type of cancer called B-cell lymphoma. Now, that doesn't mean if you've had mono, you're going to get B-cell lymphoma, but you are at a little bit higher risk of developing that type of cancer later in life if you've ever been infected by Epstein-Barr virus. That's the virus that causes mononucleosis. All right, I'm going to stop that video lecture there. The last little thing we're going to talk about are these really weird microbes. They're not really viruses, but I'm going to talk about them here now as this part of the course. And they're called prions. And that will be the last little video lecture as a substitute for our Monday that we missed.